Hello. So today we are going to discuss how we now get from the DNA and the genes to the actual proteins that make us up. So if you are watching this in class today in January in 2024, you guys just did your baby face labs. So you flipped coins and made your baby faces. And then after you made your baby faces, you physically drew what those babies looked like. That process of flipping to get what your baby is going to look like and then physically drawing it is basically modeling that process of going from a DNA to a protein. So, stuff can happen in that process. You can have mutations. You can do a lot of other stuff, but that is basically the process extremely simplified. So, what is actually happening in our cells is all of the DNA in your cells is in the actual nucleus of the cell. The reason why we keep that DNA in the nucleus is because it is extremely fragile and it is also extremely important. The reason why it's important is because it's the code for everything in our whole body. So we need to keep that safe in our nucleus. So, in order for us to make the proteins, which is the job of the ribosome, which is going to be a very important organelle that we are going to be talking about in this unit, those ribosomes are outside of the nucleus. So, to get the message, we need to turn that DNA code into a molecule of something called RNA. It leaves the nucleus, so we see it leaving through this little door right here. It leaves and then it codes for a protein, which is really in this picture. In a lot of images, you'll see of this little spheres that are these amino acids. So when these amino acids link together, they form a big chain, and that is our protein. So we're just going to learn about this process. So once again, when we're talking about this, the DNA is home. The DNA is a molecule that stores all of your body's genes. So the genes in that molecule of DNA code for all of your different traits, and not only your physical traits, but also other traits that we don't really think of. Are you lactose intolerant or not? Do you have the enzymes to be able to break down gluten that would make you have celiac disease or not? Do you have functioning beta proteins? If you don't, you have type 1 diabetes. So it codes for all of these different traits and the proteins that make up all of the different parts of you. That DNA is stored in the nucleus. And once again, it is stored in that nucleus in order to protect it because it is very, very easily degraded. So that word degraded means broken down. Some stuff that can break down DNA are heat. That is why having a fever is such a big deal. And UV light from the sun can also actually break down your DNA. And just stuff that's in your cells can break down and digest your DNA. So ribosomes are the actual organelle that makes proteins, and these are located outside of the nucleus. So we have to get that DNA message from the nucleus to the ribosome. And with this as well, every single strand of DNA that we have codes for like thousands and thousands of genes, but we only need one at a time. Like if you needed to express the gene to make more hair protein, like to grow your hair, but you express genes for every single thing in your body, that would be using a ton of resources. It would be really, really anti to what we're trying to do. So we only need to make one gene at a time. This is where the process of transcription comes into play. So we have two main processes in this: transcription and translation. The way that I always remember which order they went in when I was in high school is because they're alphabetical. Transcription, that word comes before translation. So transcription, this is the process that happens when something turns on in our body that says, hey, I need to make more hair proteins. So we are then going to copy just that gene for making hair proteins, just that gene for keratin, which is the hair protein. Okay. So that is copied in little complementary pairs, and it gets copied into a molecule of mRNA. Okay. So how this works in normal DNA when DNA is across in its double strand, so we call DNA a double helix because it's kind of shaped like this, right? We've all seen pictures of DNA before. So DNA is kind of shaped like this. On one side of our DNA, we have one letter base. So it's either an A, T, G, or C. So we'll just put like an A right here. Across from it, on the other side of that DNA molecule, we have another letter, and they are complementary to each other. So A always happens to match with T. In normal DNA, A is always going to be across from a T, and a G is always going to be across from a C. So when we are doing this, we have one unique thing that actually happens. In RNA, a T is still going to be across from an A C is still going to match with a G, G with C. However, A matches instead of with a T with a U. This is the body's way of knowing the difference between those molecules. So instead of a T in RNA, we have a U. T, once again, is still going to match with A, and G is still going to match with C. Okay. In the process of translation, that new molecule that we just made, the little letters that flew in, those are going to move out of the nucleus to the ribosome. So we see that little mRNA, the little fettuccine noodle going out and going to the ribosome. This is the process of translation. So this RNA is read by a ribosome in sets of 3. So it's like a sentence of 3-letter words, like the cat ate the rat. Okay, so we read it in sets of 3. Each of these sets of 3 is called a codon, like a code, right? So it's a codon. And every single letter set of 3 codes for one amino acid. Okay. Every single set of 3 codes for one amino acid in that growing string of protein. So the way that we do this is using this amino acid chart that we have down below here in the corner. Okay. So this is a set of 6 bases of DNA. So we are going to read them in 2 sets of 3. My first set of 3 codes for serine. The way that I'm going to actually figure that out is with this amino acid chart. My first letter is an A. So I'm going to use this column over here. So I know that the amino acid I'm looking for has to be here. My second letter is a G. So second letter is up on top. So I know that it has to be in this row and this column. And then I have AGC. So AGC is right here. So that is serine, S-E-R. Okay. So my next one that I have is tryptophan. And the way that we found that out is U. So I know it has to be in this first row. My next letter is A. So now I know it has to be in this box. UAC, that codes for TYR, which is the amino acid tryptophan. So that's how we go and code for everything. Something that you'll notice here is we also have some stop codons. So we actually have 3. Okay. So there are 3 codons that code for stop. That's basically the period at the end of the sentence. When we get to the end of the gene, we're going to see a code that is either UAA, UAG, or UGA, and that means that we are done making that particular protein. Okay. So with this, we also have some mutations that are possible. So the order of the amino acids that we have actually determines the physical shape of the protein. The shape of the protein means that it's going to be able to do a certain job. I like to think of this with shoes. So if we have like a high heel, oh, actually, hang on a second, pause. I'm back. So with Movie Magic, I got a pair of cross-country spikes. So we can see that the spikes on the bottom of the shoe have a shape for a particular function. So cross-country spikes or like track spikes, their job is to help you actually physically dig in to the ground so you can hopefully go faster, have better traction, whatever. The shape determines the function. So if we change the shape, or if we had a mutation where we took these spikes out and we're racing with them, and maybe it was really slick outside or something like that, the function of my shoe is going to change and be a little bit different. The form follows the function. Same thing with like high heels. So if you have the high heel, you have a mutation where that heel gets taken off. The job of that shoe is going to change. It's no longer going to support you, and it's going to be like a regular flat shoe. So when we have mutations, they can cause the amino acids to be in the wrong order or something like that. And that can actually change the protein. So a really, really cool science fact about this is that scientists think that there was like one single mutation thousands and thousands of years ago that caused blue eyes. And that has since gone into our population, and everybody with blue eyes somehow is descendant from that one person that had a mutation for blue eyes. So, kind of cool little science fact. We have a couple different types of mutations that we can have. Oh, gotta go back. So, a couple of different mutations that we can have. We can have substitution mutation, and this is where we switch out one base for another. So, for example, maybe the cat ate the rat becomes the cat ate the bat. So, we switched out one letter. Sometimes with these mutations, it could still code for the same amino acid or something really close, so it really doesn't matter. Not all the time, but sometimes. We can also have a mutation that is a deletion mutation. This is when we completely take out a base. So the cat ate the rat becomes the atatathurat. The reason why is because we have to read, remember, our mRNA in a set of 3. So even if we take out a letter, we now have to shift everything over. That's why sometimes this mutation is called a frame shift mutation, because we have to shift over what we're actually reading. The last type of mutation that we can have is an insertion mutation. So the cat ate the rat becomes we doubled our C, kakatat. I probably just summoned a demon to somebody's classroom by saying that, but we have to shift our reading frame over. So when we added in that extra letter, everything got shifted over, and it's going to cause a mutation downstream. So with this, we can also have a missense mutation. So this is a mutation where we are coding for a completely different amino acid, and that could change the shape of the whole protein. A nonsense mutation is if we code for a premature stop. Okay. So a nonsense mutation is if you have a string of protein that should be like 1,000 amino acids long, and in the third one, you accidentally code for a stop. You're just not even going to make that protein. Other ways that we can actually figure out which protein is which. So the one that I am looking at right now, this one is like the chart that I showed you before. We can also use this circle chart. I really, really like this circle chart for a couple reasons. I think it's just really, really easy to use, where if I have 3 bases like GGG, okay, I would start in the middle with G. I would go out G, go out one more G, and that is going to code for glycine, okay? Or if I have UAC, I would start with U, go out one A, C, and that is going to code for tryptophan, which is TYR. So a couple different things that we can do with that. So hope this helps. A little bit of background information on how we get from DNA to protein. And we will be doing a lot of practice with this, and you guys will get very, very good at it. I absolutely promise.